Today, I have the pleasure of interviewing Dr. Matt Kaeberlein at the University of Washington, who is studying the biological mechanisms that drive aging. I'm interested in staying as young as possible for as long as possible. And we know that biological aging is not the same as chronological aging. So the idea is that if we can target these underlying mechanisms, then we could essentially delay aging and delay age-related diseases. So why don't you give yourself a little introduction, what you're studying, and we can jump into these biological mechanisms that are driving aging.
Sure. So I'm Matt Kaeberlein. I'm a professor of lab medicine and pathology at the University of Washington. And I'm sort of generally interested in understanding what are the biological mechanisms of aging. And I think that brief introduction that you gave, really kind of sums up the take-home message of my research, which is to try to understand, what are the genetic and biochemical and molecular mechanisms of biological aging, with the hope that then we can actually intervene in those mechanisms to extend healthy longevity in animals and in people.
And how do we even define aging on a biological level?
Right. So, I think this is, that's a hard question to answer precisely. And I think if you ask different people in the field, you probably get slightly different definitions. I think one useful way to think about biological aging is through what have become sort of known as the hallmarks of aging or the pillars of aging. There have been a couple of reviews written on this topic. And these are really what appear to be evolutionarily conserved molecular mechanisms of aging that seem to be the processes that really drive the declines in function and increase in disease risk that go along with aging. And so these sort of fall into categories like telomere shortening or cellular senescence or stem cell exhaustion. There are 9 or 8 of them, depending on whether you subscribe to the hallmarks or the pillars view. It doesn't really matter, at least for right now, what specifically those are. I think they're sort of useful frameworks to think about aging, and they illustrate the fact that we have really learned a lot about the biological mechanisms of aging. We can put names to these things, and we understand them, at least to some extent at a fairly detailed molecular level. One of the useful aspects of thinking about the hallmarks of aging or the pillars of aging is you can also then start to think about strategies that you might take to target those hallmarks or pillars of aging, whether that's through lifestyle changes like diet or exercise, or whether that's through pharmacological approaches, drugs, small molecules, maybe natural products, or maybe even through, in the future, genetic approaches to actually change gene expression. I think that having targets is really useful, right? Because then you can actually hone in on what are strategies to maybe deal with those specific changes that we know are driving the functional declines that go along with aging. So, I sort of, maybe more so than other people in the field, really, when I think about aging from a translational perspective, or what we hope will someday be a medical perspective, I really think about the functional aspects of aging and the declines in function that go along with aging, even more so than diseases. I think we're sort of trained to think about diseases, but there are a whole bunch of functional declines that happen during human aging that often precede disease that are really important for quality of life. And so that's kind of what I think about when I'm thinking about, what are the things that I would like to change, right? I think that preventing or maybe even reversing those functional declines will be a really important component of translation, what I call translational geroscience, the application of the basic biology of aging to have an impact on health and disease.
And when you say functional decline, are you referring to like cellular declining function, or our like capability to just live our lives the way we want to?
Yeah, I think it's both, but what I was referring to a minute ago is that actual sort of more macroscopic functional decline. So, to give you a personal example, and this is, there are hundreds of these kinds of functional declines that go along with aging that we don't really ever try to categorize. So about maybe a year and a half ago, I started experiencing a lot of shoulder pain. So, I'm pretty active. I play softball every year. Well, before COVID, I played softball every year. I lift weights. I play basketball with my kids, and I started having a lot of shoulder pain. And I thought that I had a rotator cuff tear. Turns out I didn't. I had something that was called frozen shoulder or capsulitis, which is inflammation of the shoulder capsule, right? So that's something that a lot of people experience. It's extremely painful. It limited my ability to do what I wanted to do. So that's the kind of functional decline that I was referring to. And I think, anybody who's over 40, over 50 will recognize there are a lot of these things that go along with aging that have a real impact on our quality of life that, we wouldn't put necessarily in the same category as having heart failure or Alzheimer's disease, but that really impact the quality of life for lots of people. And these are driven as much by the biology of aging as Alzheimer's disease and heart disease.
Got it. Okay. And bringing up just activity and physical activity, it is interesting when you dive into the longevity research. There seems to be like different camps that are focused on protein restriction, caloric restriction, maintaining muscle mass for the functional longevity of our own physical bodies. But this changes how we view different macronutrients or how we view our diet and lifestyle. So where do you think we can strike a balance between growth hormones and activity, but also not overdoing it to the point that we might be promoting overgrowth of cells that we don't want proliferating?
Right.
Yeah. Do you want to go into that?
Yeah. So, I think that that's a really hard question to answer well, in part because almost all of the data that we have to extrapolate from comes from studies in laboratory rodents, which are really, really powerful for uncovering mechanisms and digging into the biology. But I think we have to be really cautious about extrapolating those to the human experience. And so, you talked about the nutritional studies in particular, and this is an area that I've been digging into a little bit lately. And I guess if I had to sort of sum it up in a minute, my interpretation of the mouse studies is caloric restriction is robust and usually works, but there are many instances of genetic backgrounds in mice where caloric restriction does not extend lifespan. But that's kind of the best of the nutritional strategies. And then when you start to look at things like protein restriction, intermittent fasting, fasting mimicking diet, most of the studies looking at those nutritional interventions are really caloric restriction, because the experimental groups are calorically restricted. So people sort of market them as different things, but most of the data is actually caloric restriction. When you then ask, if we look at these things like protein restriction, intermittent fasting, fasting mimicking diet in an isocaloric setting, where now they're not calorically restricted, there's really very little evidence that you get anything more than maybe a modest effect on lifespan or healthspan. And yet it gets marketed to the general public as if these are really well-established ways to slow aging. So first of all, they're not well-established in mice, at least in an isocaloric setting. Secondly, we have no idea if they're going to be beneficial in people. They might be. And I go back to caloric restriction, where if you look at the one study that looked at the effect of caloric restriction on lifespan across something like 50 different genetic backgrounds, so different mouse strains, what they found was that in about half of the mouse strains, caloric restriction actually shortened lifespan, at least at that one level of restriction that they tested. So in the only study, and there are lots of imperfections in that study, but in the only study that really tried to do this, looking at genetic variation in the context of caloric restriction, half the time it either had no effect or it actually harmed the animals from a lifespan perspective. And that's the best data, right? I would be really careful about recommending to people who are genetically diverse and environmentally diverse that they should try something that actually shortened lifespan half the time in a controlled laboratory setting. And yet, I think sometimes the perception gets way ahead of the actual data. So that's maybe a long-winded way of saying that I think that these nutritional studies in aging are super powerful to learn about mechanism. I just think we need to be careful about making recommendations on lifestyle from those, okay? So then what can we conclude? And here it gets to be a little speculative, but and this is kind of obvious, my take home is we know that too many people eat too much, right? We know that there's an obesity epidemic that leads to greater risk for a variety of diseases. And I think one way that some people in the field who like to make recommendations to the general public justify that is they say, well, any of these anti-aging diets are better than eating McDonald's all the time and being obese, which is probably true. But what's less clear to me is that you're going to get an added benefit if you are sort of normal weight and active, that caloric restriction or intermittent fasting or fasting mimicking diet or ketogenic diet, that you're really going to get a big added benefit as long as you are not overweight and obese and you're, moderately to highly active, right? That's sort of my guess. I'm not saying that you won't get any benefit. The other thing I will say is sort of my personal view on this is that it's not going to be a one size fits all. That we know based on genetic variation and environmental variation, that different people respond very, very differently to the same diet. And I don't think it's going to be the case that, a Mediterranean diet is the best diet for everyone, or a ketogenic diet is the best diet for everyone, or whatever pick your favorite fad diet, right? And so I think that there is a personalized aspect to this that we don't understand yet. The field is making progress on identifying biomarkers that might help us to get a better prognostic way to predict who's gonna benefit from different dietary interventions. But I think for now, you kind of have to figure out what works best for you to some extent. It feels kind of superficial, but really I think the old advice is still correct. Don't eat too much, exercise regularly. And then I would add to that, personally for me, what I found works really well is to really reduce the simple carbohydrates. And I, since I've done that, I feel a lot better. And so I think you kind of have to figure out what works for you, given your unique genetic makeup and the environment that you experience.
Yeah, and I want to go.
I think I also want to add, I don't know that I'm going to live much, a lot longer, and I don't know that I'm aging more slowly from that. It's just it's what gives me the best quality of life, right. And I think it's important.
Mm-hmm. And I want to jump back to the conversation on restricting in already healthy people, or like the added benefit. So do you think that caloric restriction is even extending life in half of these animal models because they're just eating less of a crappy diet? Or do you think that if they were on like a species appropriate diet that we would see these effects?
I think that's a kind of hard to answer, but my intuition is that it's not because they're eating a crappy diet. In fact, I think if you look historically over the literature in the field, mice in the laboratory today are living longer than they ever have previously, right? So I think you can make a case. I won't say that the diet is optimized for longevity, but I think you can make a case that it's not particularly bad for longevity. The other thing that is important to appreciate is, and this is again getting back to the difference between laboratory studies and the real world, right? A mouse in the laboratory in a good study, depending on the strain background, but I'm going to get a little bit technical, but let's just say C57 Black 6J, which is sort of the most commonly used mouse strain in aging studies and in biomedical research in general. Okay. So in that particular strain background, in a good study, the average lifespan will be between 800 and 900 days, depending on whether it's males or females. And so in that background, males live a little bit longer than females, but it'll be between 2 and 3 years. If you took that same mouse and you put it in the wild, or even a wild mouse and you put it in the wild, it's going to live about a year, maybe, in its natural setting. So again, I think that while the diets that are used in the lab may not be optimized for longevity, they're not obviously what we would call toxic. And so I don't see a lot of data to suggest that that's the mechanism of caloric restriction. The other point that I would make about caloric restriction is in the studies and in the strain backgrounds where you get these big effects on lifespan, and the effects can be quite large. So older studies by Rick Weindruck and Roy Walford, showed up to 60% increases in lifespan. I mean, big effects. Not only is lifespan extended, but essentially all of the diseases and functional declines that go along with aging in mice also seem to be delayed or even completely prevented. So it doesn't seem to be the case that caloric restriction is just fixing one thing, and that's why the mice are living longer. It really seems to be the case that it's slowing down the biological aging process. And that's why I say I think these dietary interventions are really useful for digging into mechanisms. And we've learned a lot about the pathways, the genetic pathways or molecular pathways that modulate aging by studying things like dietary intervention or protein restriction. I'm just less convinced that they're going to be particularly useful strategies for significantly enhancing health span and longevity in people. And that's if you could even get people to do it, right? I mean, I think that's the elephant in the room is there will be a very small percentage of people that do caloric restriction or intermittent fasting or fasting mimicking diet or ketogenic diet. But among the general population, there's really zero chance that that any of those are going to be something that become widely practiced, at least in my view. And there are a variety of reasons for that go far beyond, what we could cover in this conversation. But I just don't think it's pragmatic to really think that you're going to have a big impact on public health by, trying to market these anti-aging diets.
Mm-hmm. And by focusing on a single mechanism, do you think that could kind of backfire by like focusing on growth factors and mTOR activation or IGF-1 and then restricting protein and then becoming sarcopenic? Like there's got to be.
Yes, certainly you can push it too far. I mean, and this is the other point that I think is that I like to make about diets that people don't usually connect, which is that, diets are the same as drugs in a lot of ways, as is exercise, right? These things have real potent biological effects, and there are dose responses to these things, right? We know that too much exercise is harmful for you. We know that. We know that caloric restriction to the extreme will kill you, right? So I think it's useful to recognize that these lifestyle interventions have dose response profiles just like a drug does. And there is sort of by definition an optimum for whatever you're looking at. If lifespan is the readout, there is an optimum for your intervention for lifespan. Figuring out what that optimum is the hard part. But it is also the case that if you push it too far, you are going to, not live as long. And so absolutely, I think it can backfire at the individual level when people are trying to, combine all of these things, that you could certainly end up living shorter than you otherwise would based on your sort of lifestyle changes that you're making. The other point that I sort of have conceptually, come to recognize as important and try to communicate is, in some ways, drugs are actually maybe a better option than dietary interventions. And I say that because, at least with some drugs, we know very specifically from a biochemical perspective what they do. They target one protein, and we can titrate that in just by changing the dose of the drug that we give. Something like caloric restriction is hitting hundreds or thousands of proteins in the cell, right? So if you think about caloric restriction as a drug, it's like the dirtiest drug that ever existed. And so what that means is there are two sides to that coin. One is that it gives you the opportunity to hit multiple pathways and have bigger positive effects. The flip side of that is it gives you a lot of opportunities for really, really bad side effects. And so I think conceptually, it's just worth appreciating that fact that, diets are not safe. People who tell you that this diet is safe, they are lying to you, or they at least don't recognize that these things have side effects. And you need to pay attention to that. And if you push any diet too far, it's going to lead to bad outcomes. And so I just think it's important for people to recognize that. And a lot of people don't, because we're sort of trained to think of drugs, as dangerous and diets and exercise as safe. And all of these things have cost benefit ratios that you need to think about when you're considering, practicing them.
Totally. And I think you're referring to rapamycin mostly when you're talking about these drug interventions.
Well, that's what I think the most about, in part because that's, what I've studied for a long time. But I also don't want to suggest that there aren't other pharmacological interventions that other people are studying. So there are a variety of different molecules, some of which are prescription drugs, like metformin is another one that gets talked about a lot in the aging field, or natural products like NAD precursors, or things that are sort of in clinical development like senolytics. I think there are multiple small molecule strategies that people are taking to try to target the hallmarks of aging or the biology of aging. Rapamycin is a specific example of a small molecule that is an extremely clean drug. As far as we can tell, it's got one biochemical target. That's the only thing it really hits in the cells. And we know a lot about that pathway. It's not to say that rapamycin doesn't have side effects. At high doses, it absolutely does. But it's very clean from the perspective of biochemistry, unlike something like caloric restriction or even metformin, which still people don't really know what the mechanism of metformin is. There seem to be 4 or 5 mechanisms that are out there that might account for its ability to enhance mitochondrial function and protect against diabetes, but the reality is people don't really know. So I think even among drugs, right, there are degrees of specificity, and that's where this whole idea of a dirty drug comes from, right? Some drugs, they just don't have one target or one mechanism.
Mm-hmm. And when it comes to just our day-to-day living, how much value do you think there is on glycemic control in aging? Like, does glycemic control really influence the mechanisms that underlie aging?
Yeah, so I mean, I think that's clear in mice, absolutely, that insulin sensitivity and ability to maintain glucose homeostasis is at least strongly correlated with lifespan. I would say it's not 100% causality has been proven, but the correlation is quite strong. And I think in humans, this is where it gets a little bit harder to really make definitive statements about aging, right? So we absolutely know that if you fail to maintain glucose homeostasis and you develop diabetes, you are at greater risk for a variety of other age-related comorbidities, and you're unlikely to live as long as somebody who doesn't have diabetes, right? Is that accelerated aging, or is that something else? I think it's harder to know. But certainly, I think if we don't talk specifically about the biology of aging and we talk about life expectancy and health span expectancy, it seems to be critically important in humans as well, right? And I think from a biological perspective, it makes a lot of sense, because one of the things that has come out of studying things like caloric restriction or protein restriction is the sort of fundamental importance of nutrient sensing and nutrient response pathways in regulating biological aging. So one way to think about this is in the laboratory, the way this evolved is, people studied caloric restriction and protein restriction, and they realized that under those diets, when the mice were living longer, you had decreased growth hormone signaling and decreased IGF-1 signaling and decreased mTOR signaling. And then people started studying genetic models of those pathways, where instead of being accomplished through a diet, the genotype of those mice was changed so that growth hormone signaling was decreased, and all of a sudden it was observed that those mice started, they lived longer, right? So that's a nice confirmation that you can go from a diet, figuring out what the diet does to biology, to then a specific biological change being sufficient to give you the outcome that you're looking for, which in this case is increased lifespan. And in the case of growth hormone, IGF-1, mTOR, what adds additional confidence to that model is that it's true not only in mice, but if you look in invertebrate models like fruit flies or nematode worms, turning down those pathways slows aging, even increases lifespan to an even greater extent. So then you can start to make the argument that, well, this mechanism of nutrient response and growth signaling is conserved over, 1 billion years. There's a pretty good chance that that's going to be at least somewhat conserved if we go from a mouse to a human, which is evolutionarily much closer. So, I think that sometimes it's very, very difficult right now to make definitive statements about human aging and interventions that will impact human aging. I sort of fall into the camp that I feel pretty confident that things like growth hormone signaling, IGF-1 signaling, mTOR, will play a role in human aging, in part because there's some human data consistent with that, correlative but consistent, and also because of this evolutionary argument. It just seems, absurd from an evolutionary perspective to think that these things will be conserved over, so much evolutionary distance, and then all of a sudden it's going to be fundamentally different in people. And I also think that if you just look at aging, not from necessarily even a biologist perspective, but, the average person, if you look at an old mouse compared to a young mouse, or an old dog compared to a young dog, you're going to see many of the same changes that you see if you look at an old person compared to a young person. So even at that level, right, we sort of intrinsically know that there are biological similarities to aging across all of these different mammals. So it seems quite likely that the pathways that regulate those biological changes will also be shared.
What's your favorite way of defining biological aging? Like there's the pheno-age, there's Horvath's clock, there's different ways that we can measure. What do you prescribe to, if you do?
Yeah, so the first thing I'd say is I think all of these clocks that are being developed are a step in the right direction. I think that this is where we need to go to be able to predict which interventions are likely to have positive outcomes, particularly from a personalized perspective. And this gets back to what I was saying before, which is that, in a laboratory, we can control the environment, we can control the genotype, we can do experiments. You can't do that in people, right? So you need some other way, some other biomarkers, right, to predict whether a particular intervention and what dose of that intervention will have positive outcomes in people. So I think these clocks and other biomarkers that are being developed are the right direction to go. I will also say that I think it's premature to really try to use them in a clinical setting to make prescriptive recommendations. I don't think they've been validated in humans yet to the point where you should use them to tell somebody to do something or to stop doing something. So I think it's it's getting there, but it's not quite ready for prime time. If I had to take an approach, the approach that I like in mouse studies, and it's done sometimes in people, but not really to the same extent, is again, to really look at the functional measures, right? How well is that individual functioning as they're getting older? And in mice, you can do this, and people have really started to develop, a variety of measures to look at different aspects of frailty, right? So some of these are obvious, right? How active is the animal? How is its cognitive function? And then you combine that with molecular measures or blood chemistry measures, right, which we think reflect health. So I think those approaches are really where we should be looking over the next 10 years in humans. Is can we come up, and I think we already have this to some extent, but can we do better at coming up with a collection of functional and molecular measures of aging and those molecular measures of aging should reflect biological function, right? So the problem with the clocks, I think, especially the epigenetic clocks right now, is we don't know what they mean. We know these are marks on DNA that change with age, but from a mechanistic biological perspective, we don't know what they mean, whether they're causing anything that has anything to do with aging, right? Whereas these functional measures, like blood urea, right? That's a pretty good measure of kidney function. We can kind of correlate these things with organ and tissue decline that we know go along with aging. So I think combining those kinds of measures with actual functional measures of how well is that individual doing is really where things are going to be at for the next 10 years. And I think you can do clinical trials to figure out whether your intervention, particularly if it's something that can reverse the functional declines, whether your intervention can have a real positive effect on at least health span. Lifespan is a lot harder to do in people, right? Because people live so long. But I think we've got a real shot at validating interventions that can impact health span in people by looking at these functional measures. That's the other point that I'll just make, is one of the really exciting things in the field, over the last 5 to 10 years, has been this recognition that a lot of the interventions that we study don't just slow down aging, right? I think historically, and I certainly would have fallen into this camp, 15 years ago, historically, people always thought, well, what we're going to do is we're just going to slow down the damage that accumulates, right? What we've seen is that, at least again at a functional level, some of these interventions actually reverse the changes that go along with aging. And that's super exciting. Obviously, from a biological perspective, the mechanisms there are really interesting. But from a translational perspective, this is really important because it suggests you might not have to intervene before people have started to experience the functional declines of aging. You might actually be able to reverse them, at least some of them. And so I think that gives optimism that from a clinical trials perspective, it should be feasible if we can actually take something that's not working as well as it used to and make it work better. You should be able to detect that in the context of a clinical trial, and that's really important for the field to move forward, is to have some wins in clinical trials.
So interventions would be administered as a function of age then. So for myself, like I'm 25, but I'm thinking about aging very
Right.
early. So my priority should be slowing and delaying. But maybe midlife, that's when I would consider things that might reverse or further delay the onset of different mechanisms of aging. And in the rat models and different model organisms, most of these interventions are administered midlife, right?
I think that's been a change again over the last 5 years, really. So if you look at the literature from 10 years ago and before, almost everything was administered starting, at 6 months of age or even earlier. I think it was only with the discovery that rapamycin could be started in middle age that people really started feeling like this is possible and something that we should really be looking at. Again, because from a translational perspective, that's kind of where you want to be, right? At least if you're thinking about moving things into the clinic. I'm not saying it's not important or useful to start targeting aging at an earlier age, but if you really start thinking about moving things into the clinic and how you go about doing that, it's much easier to imagine a pathway that starts with older people, right? So it's really, that's a pretty new thing in the field that people have started maybe thinking about mid-age interventions first rather than young age interventions. But yes, that seems to be where the field is going, and I think that's a good thing. But it's not exclusive. If you look at the papers that get published, or even like the NIA interventions testing program, which tests compounds as small molecules in mice for effects on lifespan, their standard approach is to start at 9 months of age, which is a pretty young mouse. And it's only after something works starting at 9 months of age that then they will consider going back and starting at 18, 20, 24 months of age, which is when you start to hit the 50 to 65-year-old range biologically, we think, in people. So it's still not as if everybody is only testing things in middle age in mice. I think there's actually a pretty good mix of both strategies.
Gotcha. And I want to circle back to rapamycin and ask how it compares to the ketogenic diet. So I think there's only 2 studies that have shown that the ketogenic diet extends lifespan in mice. And I think that they're both administered midlife as well. But I had a question about global versus tissue-specific inhibition of mTOR, because in the one study by John Ramsey and Keith Barr, it was tissue specific. So in the liver, mTOR was inhibited, but in the muscle, it wasn't. But rapamycin globally inhibits it, right?
It does, although I think, again, there are different tissue uptake and pharmacokinetics, right, in different tissues. So certainly when you first give rapamycin to a mouse, as particularly at a high dose, you will see mTOR inhibition pretty much everywhere where you look, right? But the rate at which mTOR signaling returns to baseline is different in different tissues. So it's still not, and that, the other thing I would say about the ketogenic diet studies is I suspect if they looked more carefully, that it would be possible to see effects on mTOR in muscle and probably in brain and other tissues as well, right? So the thing about mTOR signaling that I think a lot of people don't appreciate, and I'm not at all trying to diss either of those papers. I think those are really important papers. But people who don't study mTOR signaling don't always think about is the way you do the experiment and when you actually measure mTOR signaling compared to when the mice last ate can cause you to get completely different outcomes. So if you look at a mouse that's fasted compared to a mouse that's fasted and then refed and then 4 hours later, if it's eating exactly the same diet, you will get dramatically different measurements of mTOR signaling. So I would be surprised if a ketogenic diet at some point does not reduce mTOR signaling in pretty much any tissue where you look, right? So I think we just have to be a little bit careful. I absolutely agree that tissue-specific effects on mTOR biology, and this is extendable to other pathways that affect aging, are really important. I think it's really hard to quantify those tissue-specific effects well in mice. So again, this is where I think it's useful to have those sort of tissue-specific measurements of biological activity, mTOR signaling, for example, but you need to couple that with functional measurements during aging, right? So there is this misperception out there that inhibition of mTOR with rapamycin is problematic for muscle growth and muscle maintenance during aging. I say it's a logical misperception if you don't know the literature, because mTOR activation is required to make muscle. But if you actually look at the literature, mice or rats that get rapamycin have preserved muscle as they get older, right? So that the actual data are clear that rapamycin does not impair muscle growth or muscle maintenance during aging, and it actually makes it better. So I think you kind of have to compare those, functional and global phenotypes with the molecular data to really try to understand what's going on. And so with ketogenic diet, again, and I think that the ketogenic diet literature in mice is still sort of in its infancy. Like you said, there's really only 2 studies. They both started at middle age. If you really dig into those studies, you will note that starting earlier in life actually shortened lifespan. And you will also note that in one of those 2 studies, it was not a continuous ketogenic diet. It was a cyclic ketogenic diet, right? And the continuous ketogenic diet in that study did not extend lifespan. I think it also, in the study that used the cyclic diet, I think the continuous also shortened lifespan in that study. So even in those, the only 2 studies that really have looked at lifespan of a ketogenic diet in mice, they're not completely consistent with each other. And so I think we need, again, more research in that area to really understand, why that's the case, and what's actually going on in the context of a ketogenic diet. And so again, I think if you were going to, and I'm not saying you should, but if you were going to extrapolate from those studies to your own lifestyle, right? I would look at that cyclic ketogenic diet study where the continuous ketogenic diet didn't extend lifespan and might have actually shortened it, and say, well, maybe you don't want to do a ketogenic diet for the rest of your life if you're going to do it at all, right? So again, I think that the diets are super valuable for learning about mechanisms, and one of the really interesting areas in the field is what is the role of ketone bodies in metabolism and metabolic changes during aging, and is it possible that you could get some of the benefits from caloric restriction or intermittent fasting or ketogenic diet from supplementation with ketone bodies? We don't know that yet, but it's sort of that's the way that these diets can be used to suggest mechanisms that then can be tested experimentally. And ketosis is a common biological phenomenon of many of these anti-aging diets, as is reduced mTOR signaling, right? So I think that by looking at what seems to be similar across these different diets that can extend lifespan in certain genetic backgrounds when done a certain way, that can tell us a lot about the biology of aging.
And when it comes to lifestyle interventions that have tangible takeaways, what are your views on like meal timing or macronutrients? Do you prescribe to a ketogenic diet? Are you a fan of it? Like cyclical, as you mentioned, maybe not chronically.
Yeah.
What are your thoughts on intermittent fasting and ketogenic diets?
Right. So I guess I would say, first of all, my approach is to, and this it's sort of a lifelong continuing thing, right? I mean, I think you learn as you go, right? But my personal approach is to go with what works for me, what makes me feel good, gives me lots of energy. So I don't try to adhere to a ketogenic diet, right, in any strict way. I tend to eat very, what most people would consider a pretty low carb diet, and I avoid simple carbs for the most part. And I find that when I do that, I don't have any desire to eat donuts. There's a box of Krispy Kreme donuts down in our kitchen now, and that I'm not even tempted, right? So that works for me, right? And I feel good, and I have lots of energy, and so that's what I sort of stick with. But again, because I'm really hesitant to extrapolate from the mouse studies to human aging, right? I don't try real hard to practice any of those things. Again, I think not being overweight, resistance training, I think is really important, but exercise in general and a variety of different types of exercise is really important. And other obvious things, don't smoke, don't drink too much, easier said than done sometimes, right? But so I think those are the things that I try to do. So intermittent fasting, if it works for you, great. I'm not a big fan of fasting. And again, this is going back and even looking at the mouse data, right? So if you look at the intermittent fasting data for aging in mice, it's 99% caloric restriction, right? There are very few good studies that do isocaloric intermittent fasting. There's like 2, maybe. And the rest are calorically restricted animals. And the same thing's true with the fasting mimicking diet data, right? Which has gotten very popular. Walter Longo's a good friend. I have a lot of respect for his science, but that's really caloric restriction by and large. So if that's a way that people can, lose weight, or it has clinical application, fantastic. But I'm not convinced biologically that it's different from caloric restriction. So that the time restricted feeding is a good example too. There's one study that I know of that in mice looked at a time restricted feeding what they called isocaloric, but if you actually look into the data, it's not. So this is kind of funny, right? And this is where I think you really have to. It's hard for the general public to appreciate because they don't dig into the literature, right? It'll be obvious whose paper this is if you go and look, and I'm sure if they ever watch this video, which they probably won't, they will know what I'm referring to. But so if you read this paper, right, they refer to it as isocaloric early on, but then if you keep reading, they say the mice that were on the time restricted feeding, they didn't call it time restricted feeding, that's what it was. The mice that were on the time restricted feeding diet ate significantly less than the mice on the control diet, which means it's not isocaloric, right? I mean, yeah. Those two things are completely incompatible. So it was almost isocaloric is the way I interpret that, but statistically significantly not. So that's the only real study I know of where a true time restricted feeding in the context of aging was done pretty well. And those mice lived about 10% longer than the mice that were allowed to eat continuously, compared to the mice that were calorically restricted, which lived 30% longer. So you might conclude from that it's possible to get a small increase in lifespan from time restricted feeding, but it's nowhere near as beneficial as caloric restriction.
And going back to our introduction to this conversation, maybe these interventions are not even going to benefit someone who's already has a good body composition, who's already active and eating a very whole foods based diet. So we don't even know.
And I would say, yeah, I mean, I think that's absolutely true. And the other thing, and this is a guess, right? I have no idea whether I'm right, but my guess is that even if they do benefit those people, it's going to be pretty incremental, right? So it's not going to be, the 50%, 30%, even probably 15% extension of healthy lifespan that you see in a mouse. If you're already, active and not overweight, and you have pretty good glucose homeostasis, right? My guess is that these sort of extreme intervention, I think of caloric restriction as pretty extreme. These sort of extreme interventions are not going to have a large effect on your overall health and longevity. They might have a small effect, but I would also guess that there's at least an equal chance they're going to shorten your lifespan, right? So again, this is where the cost benefit analysis, should come into play, right? We don't know. There's a chance that it might make you live longer. There's a chance it might make you live shorter. And different people who read the literature might come to different opinions about, where that cost benefit falls. I know many of my colleagues who practice intermittent fasting mimicking diet, ketogenic diet, right? So they've come to a different decision about where that cost benefit falls, and that's fine. But I think you should do that cost benefit analysis. And again, my concern with people in the field making recommendations to the general public is the general public is not, by and large, informed enough to really make those cost benefit analyses. So they rely on us, the experts, to help them. And I worry that a lot of what gets presented to the general public, is not based on a really rigorous and thoughtful cost benefit analysis. It's more based on, what I study and wanting to get personally famous, to be honest with you. So I have concerns about that. And I already talked about how caloric restriction shortens lifespan in half of the genetic backgrounds in mice where it's been tested. That's just a fact, right? And again, there are problems with that data. It was only one level of caloric restriction. So we don't know whether that's generally true, but that's the data. And so to go from that to making recommendations to the general public that they should calorically restrict, not many people are actually making that recommendation. But to do that seems a little bit irresponsible to me. So that's partly why I don't do that, right? I try to say, okay, this is what I do and it works for me, but I don't try to make general recommendations because I don't think we know enough at this point about how to slow aging in people.
Mm-hmm.
It's also why I don't tell people to take rapamycin. I mean, we don't know enough yet.
Right. I don't think anyone would even be able to comply to caloric restriction anyways, for long term.
Actually, there are quite well, yeah, I mean, I don't know. I know
than
I
Roy
couldn't,
Walford.
Yeah, right, who wrote a book called The 120-Year Diet and, died at 80
79.
70 something. Yeah, which is terrible. I mean, again, Roy Walford was a brilliant scientist and he did foundational work that sort of set the stage for where the field of aging research is today. But he also, took this other direction to try to popularize caloric restriction, which, as you said, didn't really work because most people aren't ever going to do that. But when you write a book, that has the word 120-year diet in the title, there's just no way to justify that scientifically, right? I mean, it's an attention grabber, and that's all it is. And so, I should stop talking because there are people who are writing books now with similar titles. But I mean, I think it is it's questionable from a scientific legitimacy and rigor standpoint.
So how should people, I put myself in this category, general public, how should we approach this type of material? And yeah, like what are your recommendations for people to be able to make well-educated assumptions based on what we read?
That's really hard. So I think as a general rule, if you hear somebody claim that something has been shown clinically to slow aging or extend lifespan in humans, aside from, like people who have disease, right? But normal aging. If you see people who make claims like that, your bullshit radar should come on. Sorry, I don't know if that language is okay in your podcast. You can edit it out. But it's just not true. There is nothing that has been clinically shown to slow aging in people. And so individuals who make those claims either don't know that that's the case, that it hasn't been shown clinically, or they know and they are lying to you. And either of those options is not a good option for taking advice from those people. So that would be one place I would start. And then I think, as I've said, a lot of people, probably maybe even the majority of Americans, could benefit from eating less, right? Even if they eat exactly what they're already eating less of it would be a benefit. So I don't want to say that there isn't benefit to trying some of these dietary interventions and seeing if it works for you, right? And if you can stick with it. I just wouldn't count on aging more slowly and living longer because of it. And the other point that I've already made is there is a potential risk or cost associated with any of these things. And it might very well be the case that sort of yo-yoing is worse than not doing anything at all, right? So my concern would be, if you try some of these dietary interventions, you lose a bunch of weight, you do that for 6 months, and then you stop and you gain all that weight back, and then you do it again. Is that a net positive for long-term health? And I think there's evidence in the epidemiological literature that it's probably not, right? That these cycles of large weight loss and weight gain, have adverse consequences on some of your organs, and that can actually limit lifespan. You might be, and this is the other thing I think people have to appreciate is, you might be targeting these pathways that affect aging. You might be aging more slowly. That doesn't mean you're going to live longer, right? So all you need is one organ to fail critically, especially if it's an important one like your kidney or your brain, and you're not going to live longer. Even if your body was aging more slowly, if you break one critical component, you're not going to live longer. And so I think we also have to be careful about that if we're going to make recommendations to people, we don't want to make recommendations that are going to dramatically increase their risk of dying earlier, even if they're aging more slowly. So it's
Mm-hmm.
complicated, right? And that's why it's hard. It's hard if you really want to be thoughtful to make general recommendations in this area right now. Again, I think as we learn more, particularly as we start to get better biomarkers, that there may be opportunities to do a better job of being able to make recommendations to people. But it just feels a little bit premature to me to really be strongly encouraging people to make lifestyle changes based on the sort of current state of knowledge in the field.
So then what should the goal be? So what should we just be trying to optimize body composition, maybe more muscle mass, less fat mass, and then maintaining insulin sensitivity? Are those good markers for good mitochondrial function, less or better immune system? What could be
Yeah,
our
so
goals?
right. So I think it depends on the person, right? And it also depends on your access to healthcare and sort of even more advanced approaches to looking at a panel of different potential biomarkers, right? So if you're talking about the average person that may or may not have good quality healthcare, they may not be able to even think about following their insulin sensitivity and glucose homeostasis closely, right? And so I think for the average person, right, it needs to be sort of very general recommendations, which we've sort of already covered. And it's the ones that by and large, the medical community already encourages people to do. It's just that we have social and public policy forces that are acting at odds with that, which is why we have an obesity epidemic right now. But for people who have the interest and the ability to do more sophisticated sorts of biomarker analyses on themselves, yeah, I mean, I think that absolutely. So looking at things like insulin sensitivity, your fasting glucose levels, your postprandial glucose levels, comprehensive blood chemistry, and functional measures, right? We've already talked about some of the functional measures of aging. That's something that I actually don't see a lot of. There's this whole community of people who self-experiment, right? Who take the latest supplements, often supplement stacks that target every possible, protein or pathway that's ever been implicated in aging. And again, I don't have any problem with that. Most of those people are pretty knowledgeable, or at least, knowledgeable lay people. So if you want to self-experiment, great. That's up to you. And some of them do sort of do these sort of more sophisticated biomarker analyses on themselves, but not very many of them, at least that I've seen, really also then try to look at their functional aspects of aging, right? Muscle function, cognitive function, immune function. Those are not as easy and obvious for how you quantify those well. But geriatricians know a lot about the way that humans age, and they've developed assays that can sort of track those parameters. And so I think those are useful for people who are really interested in sort of experimenting on themselves or trying to have the best outcomes during aging that they possibly can. Now, the challenge is for somebody like you, right, is that you're functioning pretty darn well. So how do you, it's harder to see changes or delays that are meaningful, right? So I think it's much harder for people who are younger to really know whether the steps that they're taking are net beneficial, no effect, or negative, right? So again, my feeling would be, especially at your age, be fairly cautious about what you do. Don't do anything too extreme, especially because there's a good chance that, by the time you get to 40, 45, 50, we will know a lot more than we know right now, right? About how to do this well. So I wouldn't rush out and start trying to make, major changes to try to live longer and healthier down the road.
Okay, but what are your thoughts on cycling in and out of ketosis, no matter what your age is? Maybe we'll say an adult over the age of 18. Do you think that there's value in going in and out of ketosis periodically?
I think there absolutely could. Yeah, well, yeah. I mean, I think there absolutely could be, and I think the data that we know about in animal studies and in people support that. So, again, this is where I'm not recommending it, but I think that the evidence that I know of that's available certainly suggests that periodic ketosis is probably beneficial and probably not harmful, right? So, but that's my cost benefit analysis, right? But yeah, and again, I think part of that also is that if you are periodically going into ketosis with any sort of regular frequency, you're also probably not obese, right? So there is the public health component, which we talked about earlier, which is that any of these anti-aging diets is probably better than the typical diet of the typical American or a person in a developed country, right? So there is that component too. But yeah, I mean, I think you can make an argument that there's a good chance that periodic ketosis has health benefits in and of itself that go beyond just the reduced calories. But again, I don't know. I mean, it's just not, the data is not as strong as it needs to be to really say that with any confidence.
Mm-hmm. The way I view ketosis, especially if it's induced by fasting, is that I don't know what mechanism it might be benefiting me through, but it's a marker of a lot of different things that are going on, whether it's glycogen depletion, growth hormone suppression like IGF-1, mTOR, low insulin, low glucose. So I don't know. I think
I think you're right. I mean, I think so from that perspective, it's something that can be measured easily, right? I mean, you can get these monitors that you breathe into, or if you want to do it by blood, right, that will give you some measure of your ketone bodies. You're going to show me your monitor.
I am so one easy. Of those weird self-experimenters.
So, I think that for the reason that it's easy and that anybody who can spend $50, right, to get the monitor, how much ever it costs, I don't know how much these things cost, but if you can afford the monitor, you can do it, right? I agree with you. It's a useful way that you can get data that serves as a proxy for some of these other biological mechanisms that seem to be important, again, in laboratory animals. I think you can make a pretty good argument. You can definitely make an argument that in humans, chronic high IGF-1 signaling in particular, and chronic hyperactivation of mTOR in certain tissues leads causally, very likely at least, to many of the pathologies that go along with aging. So it makes sense that by periodically turning those things down, whether it's through rapamycin or through fasting, that it's likely to have health benefits. But there's always a but. It hasn't been proven in people. And again, what you need to be aware of are the potential costs that could go along with that. Probably, restricting your meals to 8 hours a day doesn't have any major costs, but we don't really know. And oftentimes we don't think about the non-obvious costs that sometimes go along with particularly dietary interventions and the social and also mental health costs. I think at least for some people, right, restricting your food to 8 hours a day or 10 hours a day creates a whole lot of mental anxiety. And we don't always appreciate that's a side effect, right? And it can, I would suggest in some people, lead to more severe mental health problems down the road. So I think it is important to really think about, what are the potential costs to any of these lifestyle interventions that people are considering doing, with the goal of aging more slowly, given that we don't know that these things are going to work in people.
So what I'm gathering from this conversation is that you're not confident in any of these dietary interventions, but you are confident in exercise.
Yes. Well, so again, I think it's important to ask who we're comparing to, right? And again, and I'm trying to be precise, but I think you have to be precise if you really want to get to the meat of the conversation, right? So I think we have to be clear who we're comparing to. So again, if we're comparing to the average American, I'm confident that these anti-aging diets are going to be better for most people in terms of health outcomes and longevity. I'm also confident that exercise is going to be better for most people. If you're talking about slowing aging, right? I think for exercise, at least, you can make a strong argument based on the epidemiological data that people who exercise regularly and moderately live longer and tend to be healthier as they get older, both in terms of disease incidence and functional measures of aging, right? So by that definition, it seems likely that exercise slows aging in humans, right? Exactly what the mechanisms are and the biological pathways that are involved is a little bit unclear, but it seems likely to me personally, which is interesting because in mice, it's a little bit less clear, right? There haven't been a lot of studies on exercise and aging in mice, but the ones that have been done look like exercise probably increases average lifespan of the population without increasing maximum lifespan. So what that means for aging, we don't really know. But unlike caloric restriction, which really robustly pushes the survival curve out, exercise doesn't seem to do that. Now, it's harder to do with good exercise studies in mice for obvious reasons, but that's a little bit of a disconnect between the basic preclinical literature and what we see in people. But yeah, I feel pretty confident that regular exercise is among the best, if not the best, interventions that people can take, aside from not eating too much, to give themselves the best opportunity to be healthy as they age. But I also want to note that exercise has real side effects, right? I mean, I exercise a pretty fair amount, and I experience side effects every single freaking day from exercise. Usually they're pretty mild, but not always. Like I've had not severe injuries, but I've had some pretty significant injuries. I'm missing a toenail right now because of exercise, right? So again, we don't always appreciate that exercise has side effects too. And I'm willing, I'm willing to experience those side effects, but it's not without cost. So again, I think this cost benefit analysis should come into play when you really think about the way that you want to live your life.
And when we think about the hallmarks of aging and exercise mechanistically, do we know anything about cell senescence and exercise?
A little bit. So again, the animal studies there aren't that many really good animal studies in this area. So certainly, if you think about the hallmarks of aging, one hallmark of aging is mitochondrial dysfunction. That's probably the area where exercise has clear benefits. It's clear in laboratory animals and in people that exercise can improve age-related declines in mitochondrial function, which then obviously has effects on many metabolic parameters and also on other hallmarks of aging. The other thing that exercise seems to do pretty well is to tamp down on some of the chronic inflammatory processes that go along with aging. And I think the mechanisms there aren't completely worked out, and it may end up tying back into mitochondrial function and metabolism. But my understanding is that from the literature is that there's pretty good evidence that with aging, we know that there's an increase in chronic inflammatory signaling, which is in part due to cell senescence, not completely, but is partially due to accumulation of senescent cells, and that exercise can turn that down, at least chronically, which is kind of interesting because exercise can also lead to acute inflammation, but chronic inflammation actually seems to be reduced. And that may have to do with cycling of mTOR. So again, this gets back to this idea that, in order to build muscle, I know everything comes back to mTOR in my world, but in order to build muscle, right, you have to turn up mTOR in the muscle. But also associated with that transient activation of mTOR is downregulation of mTOR following that burst of activity, right? So it may have that the effects on inflammation may have to do with the sort of cyclic activity of these growth promoting pathways rather than, their role that they're most known for, at least in the exercise community, which is promoting muscle growth. That's speculation on my part. But so to get back to your original question, I think we can certainly look at mitochondrial function and metabolism, and we can also look at inflammation, which is related to cellular senescence, stem cell exhaustion, perturbations in intracellular communication. So at least 3 or 4 of the hallmarks of aging, you can make a compelling case that exercise is positively impacting.
Okay, good to know, because I'm an avid exerciser. And I like to promote exercise for everyone.
Yeah, I mean, I think we should. But I do want to come back to this side effect issue, because this is something that I just because I think, particularly with exercise, it's useful to note. I mean, again, anybody who exercises a lot can just look at their own experience and recognize a long list of side effects that they have encountered because of exercise, right? And if you think about, if you had a drug that you were trying to get approved by the FDA to target aging, right, that was better than exercise, and you look at the side effect list that would have to go on that carton, right, the container that the drug goes in, if you put all of the side effects of exercise on the side of a box for a pill, people are going to freak out, right? There's no way you would get that approved by FDA. So as part of thinking about moving anti-aging interventions into the clinic, there also has to be this recognition and discussion about risk reward in the context of people who are not diseased, right? So older people who are functioning as well as you would expect for their age. They're not healthy. They're not healthy like they were when they were 30, but they're not diseased, but they're functionally impaired. I'm sorry, it's true. Okay, I'm going to be 50 in 2 weeks, and I will admit I'm functionally impaired to where I was 25 years ago. I think we just have to accept that. Okay, so they are functionally impaired, but they're not sick. How do you get a drug that's going to have some side effects? And I think exercise is a great one, because everybody talks about we should all be exercising, and I don't disagree with that. But how do you get a drug approved that might be better than exercise if it has the same side effect profile as exercise for use in older people of normal health status? It's a real challenge, and I think it's something that that is going to have to be addressed. And I think it will be. I mean, I think the conversations are happening, but there does have to be this shift in the way that especially clinicians think about aging in order to get them on board. And you have to recognize that a 50-year-old, a 60-year-old is functionally impaired compared to where they were 30 years ago in many, many domains. Maybe not all, but in many, many domains of function, it's the case. And so there should be some tolerance for side effect if you can actually reverse some of those impairments. At least that's my view.
So are you bringing this up not to go against exercise, but be more accepting again for drugs?
Yeah. No, I'm not at all trying to say that you should stop exercising. You may want to, like if you're experiencing really bad side effects, you may want to reconsider the type of exercise or the amount of exercise you're doing. But no, I mean, I think I'm not at all trying to diss on exercise. I'm just trying to make the point that exercise is a drug. If you think about it like a drug, and it's in a lot of ways it is, should we be willing to tolerate the same side effect profile from a small molecule if you can get the same or better benefits? And I would argue, yes, we probably should. But there is not a path to actually get there. That's the point I'm trying to make. That if we had a drug that gave you more benefits than exercise and had exactly the same side effects, there is currently no path to getting that drug to being used in people. There's no regulatory path, and clinicians wouldn't prescribe it. And I think that requires a change in thinking and a change in policy that hasn't happened yet.
That's a big barrier, though. How do you think that we'll get past that? Because there maybe are some drugs that haven't got through this type of regulatory thing that actually benefits us as the public. But when it comes to maybe some drugs that have a net positive, then these regulatory...
Yeah, I mean, I agree. It's hard. I think, so I'm pretty optimistic by nature. I think it's going to be 2 steps forward, 1 step back. I think we've already sort of seen that. There are, as I've said, a variety of people in the field having discussions with regulators at FDA. I think if we can get some of the policymakers and politicians to recognize this, that will help. I'm not a huge fan of bureaucracies, and I think there are lots of ways that FDA can be reformed to better serve the American public. And I think the way that we evaluate and regulate drugs is a mess, to be frank. So I think there are changes that can be made, and I think that those discussions are happening. And I'm not So if there's anybody from FDA listening to this, I'm not blaming you, right? It's a function of the way that the institute has been built and the bureaucracy. I think the people at FDA and the people that I've interacted with and from talking to my colleagues that others have interacted with are by and large great. And you can talk to them one-on-one, you can have discussions around this, and they recognize some of the challenges. And I think they're starting to recognize the need to reconsider the way we think about aging, at least, and the functional declines that go along with aging. But it's not going to happen right away. It's going to be hard. We're going to have clinical trials that fail. I think the first clinical trial for a drug that targets aging, even if the endpoint isn't aging, but everybody recognizes that's how it's working, the first FDA approval of a drug like that will be a big step in the right direction, and it will both be proof of principle and facilitate future clinical trials because the framework will already be there. We had one disappointment with an mTOR inhibitor called RTB101 that a company called Restore Bio was in phase 3 trials for. And I think that this may be actually an interesting and unfortunate case where the drug probably does work, but because of the way that FDA approached the clinical trial and the endpoint that they demanded, the drug did not reach the endpoint, did not meet the endpoint, and the clinical trial was halted. But it probably works. At least that's my guess. And I think we'll see that data published eventually, and I think it will at least be convincing to some people in the community that this drug probably could help boost immune function in older people. It's just because of the way that it was approached, and because of some of the changes that FDA made to the endpoint, the clinical trial was unsuccessful. The clinical trials are hard. So again, that's what I mean. 2 steps forward, 1 step back. 2 steps forward, 1 step back. Hopefully, though, the net progress will be forward. And I mean, I think it will be. We'll get there. I don't know how long it's going to take.
Mm-hmm. And so what are you personally working on right now? Or what's your end goal with your research? Where will you ever get to the point where you're like, I could die happy?
Well, I think I could die happy now. Like I said, I'm pretty optimistic.
Okay, but in regard to your research, is there any
Yeah, I mean, I don't know. I don't know that. I certainly hope that I don't ever get to the point where I feel like I'm done. I think that so it will evolve. One of the things I've noticed in my scientific career is I can't really predict where it's going to go, right? And I think that's a feature of I'm just sort of a naturally curious person, and if something comes along that I see as important, then I go for it. I don't hesitate to go in a different direction. So I don't know where I'll be, but it will probably be aging for sure. I can't imagine going into a completely different area. So I think it will be in the field of aging. I will say that over the last decade, my lab still does a lot of very basic work in invertebrate models, yeast and C. elegans, very fundamental biology. But I have certainly gotten much more interested in translational and clinical applications. And so if I had to guess, that's where I expect that my work will go in the future, is more in that direction. And both thinking about how do we move interventions from preclinical aging research into the clinic, hopefully helping, through things like this, having discussions with people at FDA, having discussions with policymakers, helping to accelerate that process, but then hopefully also being involved in taking some things from the preclinical world into the clinic. And so obviously, one of the projects that has become a much bigger part of what I'm doing in the last 5 years is the Dog Aging Project, where we are both trying to understand aging in pet dogs and also hopefully slow aging in pet dogs. The first attempt at that is a clinical trial of rapamycin. So, it's not a human clinic, but it is a clinic. This is a double-blind, placebo-controlled veterinary clinical trial. We're not going for FDA approval, but in principle, you could get a drug approved by FDA for aging using the template that we're building. And in pet dogs, you can actually use lifespan as an endpoint, which is hard to do and probably impossible to do in a clinical trial in people. So hopefully I expect that will continue to be, an area that I'm active in. And I would like to go beyond rapamycin. I mean, I feel like rapamycin is sort of our best shot right now at slowing aging in a large mammal and in people. But it's not the only one. And I think as long as you can design these clinical trials safely, right, that we should really be thinking about testing other interventions in companion dogs for exactly the reason I just alluded to. We can, as long as the study's sufficiently powered, we can know if we're slowing aging in 3 to 5 years. Like, we can really answer that question, which you can't do in humans in that time frame. It's just, I'm sorry, it's just not feasible. So I think there's a lot of potential there, and I expect that will be something that I continue to be actively involved in. And then I hope that we can actually start doing some clinical trial work ourselves for specific indications. Again, I don't think it's feasible to use, certainly it's not feasible to use lifespan as an endpoint in a human clinical trial for aging. It's hard to use age-related disease, although not impossible. But I really like the model, at least, that Restore Bio took, which is where you have a functional decline that goes along with aging, and you try to see if you can make that better. And so in their case, they were looking at vaccine response and immune function. But in principle, that approach could be applied to a variety of age-related endpoints. And pretty much, if you really just think about different organ systems, you can think about functional declines in those organs that could potentially be endpoints for a clinical trial. I'm pretty interested and excited about oral endpoints. So periodontal disease, gum inflammation, periodontal bone, salivary function, in part because people don't think about it a lot in the field. And I like to, be in thinking about things that, are different than what everybody else is thinking about, but also because it's a real problem for a lot of people as they get older, right? 2/3 of Americans have periodontal disease in their 60s. Almost all older people have declines in salivary function, which you don't think about when you're young, but it kind of sucks to have a dry mouth all the time when you're old. You get dysregulation of the oral microbiome, and we know that rapamycin, at least in mice, reverses all of those clinical features of oral aging. So that's a pretty straightforward clinical trial to think about, because dentists are really good at looking at gum inflammation and bone depth and things like that, and oral microbiome. So it's a pretty straightforward clinical trial. So I don't know if we'll be the ones to do it or if somebody else will do it, but I think that's a nice endpoint to think about for clinical trials. But that's a specific example. You can come up with endpoints for a variety of age-related indications, and I've got ideas on others. I mentioned my frozen shoulder. I actually think that there's a really good chance that rapamycin would work really well for frozen shoulder. And I will note, I do no longer have frozen shoulder.
And did you take rapamycin?
I'm just saying, I would bet that rapamycin probably works really well.
Is that because of the inflammation and rapamycin's effect?
Yep. Yeah. So when I got diagnosed, and when I went and looked and figured out what it was, and then it was inflammation of the shoulder capsule. I mean, the first thing that occurred to me is, okay, this is chronic inflammation. It's age-related. What do I know of that might be good for chronic age-related inflammation, right? So I think I think there are a bunch of age-related indications that are driven by chronic inflammation. We know that's true, right? Mm-hmm. And so in principle, these interventions that target aging, in general, they're really good at tamping down that chronic age-related inflammation, at least temporarily. So I think that there's a pretty good opportunity there. And I mean, there are other approaches, like rapamycin is one, senolytics are another that are really effective at turning down age-related inflammation. I think that there are probably multiple strategies that can be taken in terms of the intervention, but there are also multiple endpoints that could potentially be used to get to a clinical trial and get approval for your drug. And, I don't know that I will ever be the guy to go through the FDA clinical trial process. Maybe, I mean, I'm not ruling it out, but I certainly would like to help, right? And I think, coming up with ideas for what might be indications that people can go after is one way that I can help.
Awesome. Very exciting times. And I'm excited for the future of this in this field of research, but I don't want to take up too much of your time. So we'll wrap it up here. If you want to just mention where people can find you online, and we'll sign off.
Sure. So I'm easy to find online, but If you want to tweet at me, I'm on Twitter at mkaeberlein. If you're interested in the dog aging project, if you have a dog, I would strongly encourage you to nominate your dog to participate in the dog aging project. You don't have to be in the clinical trial. You can be part of our longitudinal study of aging in dogs, which is going to be super powerful and I think really important. We're hoping to get 100,000 dogs in that longitudinal study. So you can go to dogagingproject.org. There's a little button at the top, nominate, and you can join the Dog Aging Project PAC and be a citizen scientist contributing to aging research. So I definitely encourage people to do that.
That's awesome. Well, thank you so much for taking the time to chat with me today.
Sure. Been a pleasure.